Hello students, welcome to E-Pathshala. I am Dr. M. Pungodai, working as Assistant Professor in Zoology, Department of Zoology, Avanashilingam University for Women. In this module, let us have an overview on the role of liver and pancreas in the process of digestion. The digestion of food materials in the small intestine by chemical means relies on the function of liver and pancreas. Liver, the complex organ in the body, breaks down the nutrients and drugs to useful components and transport them to other parts of tissues or store them as glycogen, the energy supplier. The liver, a vascular filter, extracts waste and toxic matter from the body and acts as detoxifying agent. Liver produces bile and exports to the duodenum and acts as an emulsifying agent. The pancreas produces pancreatic juice, which contains digestive enzymes and bicarbonate ions, helps to break down carbohydrates, lipids and proteins in the body and transports it to the duodenum. It also plays a role in regulating the blood sugar levels. The liver plays an important role in the production of bile for digestion process. Hepatocytes, the working cells of liver, produce a slightly alkaline yellowish or greenish liquid known as bile, which is composed of water, bile salts, lecithin, bilirubin, cholesterol, bicarbonate, inorganic salts and trace metals. Lecithin and bile salts synthesized in the liver helps to solubilize the fat in the small intestine, and bicarbonate neutralizes the acid in the duodenum. The other ingredient denotes the substance extracted from blood by liver and excreted through bile. When bile passes through the ducts, the addition of sodium, bicarbonate and water increases the total quantity of bile. Bile secreted by hepatocytes enter gallbladder and gets stored. Bile gets concentrated in the gallbladder by the absorption of sodium chloride and water. When food containing fat reaches the duodenum, cholecystokinin is released by the intestine, which stimulates the gallbladder to release bile and helps to emulsify the larger mass of fats into smaller ones. Emulsification increases the surface area of the lipids, making them easier to digest. The bile salts combine with fats and makes complexes known as micelles. The fat ingested in the intestine are easily absorbed in the blood with the bile salts. Bile helps to neutralize the excessive stomach acid before the food enters the ileum and also acts as an antibacterial agent. Bile salts stimulates the peristaltic movement of the intestine and act as a laxative. Bile salts also prevent the formation of gallstones by keeping the cholesterol and lecithin in aqueous condition. Bile salts also help the body to dispose of excess cholesterol. When large quantities of cholesterol are ingested, they are turned into bile salts, which are reabsorbed and can be reused. However, when the body's cholesterol stores are high, bile salt reabsorption can be reduced, which causes the bile salts to be eliminated along with fecal matter. Because of the high amounts of cholesterol present in the bile, this provides the body with a method of eliminating excess cholesterol so that it does not accumulate in the blood and maintains the cholesterol homeostasis in the blood. Liver also helps in the excretion of bilirubin, a component of bile. Bilirubin, a product of liver's digestion, is produced during the recycling of worn-out RBCs or red blood cells. The red blood cells after its lifespan are destroyed in spleen and passed on to the liver, where it metabolizes the hemoglobin of RBC into heme and globin. Globin is further broken down and used as source for energy in the body, whereas iron containing heme cannot be recycled by the body and gets converted to bilirubin and eliminated from the body via bile. The other substances excreted in bile are copper, iron, toxins, cholesterol, lecithin, alkaline phosphatase and bacteria. The Kupffer cells of the liver plays an important or vital role in the defense mechanism of the body. The liver is involved in the elimination of foreign bodies such as bacteria or antigens. The Kupffer cells in the liver swallow and digest the foreign particles by the process called phagocytosis. These cells are also involved in the production of interleukins and tumor necrosis factor, which activates the immune system of the body. Regarding the role of liver in metabolism, hepatocytes, the major metabolic overachievers in the body, are responsible for metabolizing the carbohydrates into biologically useful materials. Liver, which plays a buffering role on maintaining and regulating the blood glucose level, and make use of enzymes that are alternatively turned on or off depending on glucose level. The carbohydrates are broken down to glucose in the small intestine and absorbed into the bloodstream and directly transported to the liver. Excess glucose from blood is converted into glycogen by a process called as glycogenesis, and the glycogen formed can be broken down to glucose by glycogenolysis and released back into the glucose when blood glucose level declines. When the stored glycogen gets exhausted, liver make glucose from amino acids and fatty acids through a process called as gluconeogenesis. The lactate formed from glycogen in the muscles as a result of glycogenolysis and glycolysis is carried by the blood to the liver, where it is converted into glucose by gluconeogenesis and stored as glycogen. Thus, the interconversion of liver and muscle glycogen is mediated by a cycle called as Cori cycle. The glycerol obtained from the hydrolysis of fat gets converted to glycerol 3-phosphate by glycerokinase with the help of ATP in liver and kidney. Glycerol 3-phosphate is oxidized to dihydroxyacetone phosphate, which then gets converted to glucose. Thus, the role of liver in a carbohydrate metabolism is very remarkable, and similarly the liver plays a very important role in protein metabolism. The liver synthesizes non-essential amino acids from other amino acids, glucose and fatty acids. Nitrogen containing amino groups has to be removed by deamination before they can be used as a source of energy or converted into glucose, or else the ammonia released will be toxic to brain. The liver degrades the proteins and removes the toxic ammonium ions by converting it to urea, which is eliminated with urine. The proteins are metabolized to amino acids also by transamination, which takes place in liver hepatocytes, where the amino group is removed from the amino acid and donated to a keto acid, converting that into an amino acid by transaminases enzyme. The liver transaminases for oxaloacetate or alpha-ketoglutarate plays a key role in funneling nitrogen from amino acid metabolism to aspartate and glutamate for the conversion of urea for excretion of nitrogen. In similar manner, in muscles, the use of pyruvate for transamination gives alanine, which is carried by the bloodstream to the liver via glucose-alanine cycle. The liver is responsible for the synthesis and secretion of a wide range of proteins into the bloodstream, including blood coagulation factors 1, which is fibrinogen, 2, prothrombin, 5, 7, 8, 9, 10, 11 and 13, as well as C, protein S and antithrombin, and transporter proteins such as lipoproteins and albumin. The role played by liver in lipid metabolism for controlling energy storage, as it is as capacity for liver transport, lipogenesis, lipid transport, lipid oxidation and lipolysis. The lipids are mainly stored in liver and adipose tissues. When chylomicrons, which is a lipoprotein particle that consists of triglycerides, phospholipids, cholesterol, and proteins, travel through the capillaries of hepatocytes or adipocytes, the enzyme lipoprotein lipase hydrolyzes the triglycerides of chylomicrons into free fatty acids and glycerol. The remnants of chylomicrons, namely the cholesterol and phospholipids, are released into the blood and they combine with the proteins to form lipoproteins and distributed throughout the body. The high-density lipoprotein, which carries cholesterol and phospholipids from tissues and organs, are directed back to liver for degradation and elimination. High-density lipoprotein is also used for the formation of bile in liver. The cholesterol and phospholipids present in the liver are transported to different areas of body by low-density lipoproteins. The liver also removes low-density lipoproteins and other lipoproteins from circulation by receptor-mediated endocytosis. Additionally, excess cholesterol from cell is brought back to the liver by the high-density lipoproteins in a process known as reverse cholesterol transport. Triglycerides from the liver are transported to adipose tissues by very low-density lipoprotein. The acetyl-CoA formed in the liver during fatty oxidation is converted into ketone bodies, namely acetoacetate, beta-hydroxybutyrate, and acetone, which are transported to other tissues through bloodstream. Acetoacetate and beta-hydroxybutyrate are converted into acetyl-CoA through citric acid cycle. Ketone bodies cannot be used as fuel by the liver since liver lacks the enzyme thiophorase. But acetone in low concentration is taken up by the liver and undergoes detoxification through the methyl glyoxyl pathway, which ends with lactate. Acetone in high concentration due to prolonged fasting or a ketogenic diet is absorbed by cells other than those in the liver and enters a different pathway via 1,2-propanediol, which is turned into pyruvate. Thus, liver acts as a producer of ketone bodies for all the tissues but not as a consumer. Liver also utilizes cholesterol and phospholipids to produce bile that helps in dietary fat digestion and absorption in small intestine. The liver stores many essential nutrients, vitamins and minerals obtained from the blood and releases them whenever needed in the body. Under the influence of insulin, glucose is transported into hepatocytes and stored as glycogen, a reserve food material. Similarly, liver cells absorb and store fatty acids from the digested triglycerides and maintain the homeostasis of blood glucose. Also, vitamins B, 12, A, D, E, K and minerals such as iron and copper are stored in liver and supplied to all parts of tissues whenever needed. The liver also plays a vital role as a hormone processor, manufacturing and regulating hormone levels, directing various hormones to perform their proper functions in other parts of the body and also in detoxification. The liver metabolizes hormones and other substances using two primary phases known as phase 1 and phase 2 pathways. During phase 1, hormones or substances are metabolized directly, but often they are converted into intermediate forms which are then further metabolized in phase 2. During phase 2, a process known as conjugation begins, in which nutrients such as amino acids are combined with hormones and other substances and are converted to water-soluble compounds that can be excreted efficiently in the urine or stool. Thus, in both the phases of biological transformation, liver plays the body or provides the body with nutrients and supports the excretion of excess or toxic substances in the urine, liver bile, perspiration and exhaled air. Thus, the hepatocytes are involved in the removal of harmful and toxic substances generated during nutrient metabolism by detoxification. Liver also have various other metabolic roles. Liver, along with the skin and kidney, participates in the synthesis of vitamin D, which helps in the absorption of calcium and phosphorus and also regulates the permeability of cell membrane. The enzyme catalase synthesized by liver break down the powerful oxidizing agent and toxic substance hydrogen peroxide into water and oxygen. The liver is responsible for immunological effects, and the mononuclear phagocyte system of the liver contains many immunologically active cells which acts as a sieve for antigens carried to it via the portal system. Liver also generates enormous amount of heat because of the occurrence of metabolic reactions. Liver also performs homeopoietic and homeolytic functions. The blood cells in fetus are produced by liver, and it stores vitamin B12 and iron necessary for erythropoiesis and for the synthesis of hemoglobin. Liver also synthesizes thrombopoietin, which helps in the production of thrombocytes. The hormone, namely parathormone, cortisol, insulin, glucagon, growth hormone, and estrogen are catabolized in the liver. It inactivates and converts the fat-soluble drugs into water-soluble substances which are excreted through bile or urine. The liver also produces insulin-like growth factors 1, a polypeptide protein hormone that plays an important role in childhood growth and continues to have anabolic effects in adults. The liver also produces special sex hormone binding globulin proteins termed as chaperone throughout the body. When its level is too high, the protein also inactivates the body's own hormone production as part of its chaperoning duty. The liver produces angiotensinogen, a protein hormone that causes the blood vessels to become narrower and helps to maintain the blood pressure and fluid balance in the body. It also secretes blood plasma proteins, namely the albumin, complement, coagulation factors, and globin. Albumin, the most abundant protein in bloodstream, regulates the exchange of water between blood and tissues and also essential in the maintenance of oncotic pressure and transport fatty acids and steroid hormones. Complement plays a vital role in the immune system's defenses against infection. Coagulation factors enable blood to coagulate when the blood vessel wall is damaged. Coming to the role of pancreas in digestion, pancreas, a major organ, plays a vital or essential role in converting the food into fuel for its body's cell. They perform an exocrine function that helps to secrete enzymes involved in digesting substances from outside the body, and an endocrine function that secretes the hormone and other substance within pancreatic islets and helps to control blood sugar levels and metabolism within the body. Let us have an idea on the exocrine functions of pancreas. The pancreas, which secretes pancreatic juice, contains variety of enzymes which are synthesized from the acinar cells and pours into the duodenum, which helps to break down the carbohydrates, proteins, and lipids. Along with the expulsion of enzymes, high concentration of bicarbonate was released from the pancreatic duct cells, which make the pancreatic secretions alkaline. This alkaline fluid enters into the small intestine and assists in neutralizing the acidic kind from the stomach and provides a better environment for the activation of pancreatic enzymes. The acinar cells present in the pancreas produce the digestive enzymes which are in inactive form. Once they enter the intestine, the enzymes are activated and broken down into complex food materials as to simpler ones. The pancreatic juice produced from acinar cells has digestive functions and neutralizing action. The pancreatic juice plays a vital role in the digestion of proteins, lipids, and also has a mild action on carbohydrates. The digestion of protein by pancreatic enzymes plays a very important role in breaking down the proteins. The major proteolytic enzymes of pancreatic juice are trypsin and chymotrypsin, and other includes nuclease, elastase, carboxypeptidase, and collagenase. The enzyme enteropeptidase or enterokinase converts the inactive trypsinogen into active trypsin, a powerful proteolytic enzyme. Trypsin also converts caseinogen in the milk into casein, which results in the curdling of milk. It helps in the acceleration of blood clotting and also performs autocatalytic functions, that is, once formed trypsin cannot convert trypsinogen into trypsin. Trypsin also activates the inactive chymotrypsinogen into chymotrypsin, which hydrolyzes the proteins and digests the casein rapidly. Trypsin also activates the procarboxypeptidases into carboxypeptidases, which breaks the terminal bond of protein molecules. The nucleases, namely ribonuclease and deoxyribonuclease, present in the pancreatic juice, digest the nucleic acids, that is the DNA and RNA, into nucleotides. Trypsin also helps in the activation of elastase into elastase, which digests the elastic fibers. Similarly, trypsin activates procollagenase into collagenase, which digests the collagen. The pancreatic lipolytic enzymes, which is secreted by the pancreatic juice, includes pancreatic lipase, cholesterol ester hydrolase, phospholipase A and B. Pancreatic lipase hydrolyze the glycerides into monoglycerides and fatty acids. Its activity is accelerated by bile salts, which helps in the emulsification of fat prior to digestion. Colipase, which is a coenzyme, also facilitates the hydrolysis of dietary lipids. Trypsin activates the inactive colipase into active colipase. Phospholipase A is activated by trypsin, converts lecithin and cephalin into lysolecithin and lysocephalin, which in turn is acted by phospholipase B to yield phosphorylcholine and free fatty acids. Cholesterol ester hydrolase cholesterol ester into free cholesterol and fatty acids. The role of pancreatic amylase on carbohydrates is the enzyme alpha amylase splits the alpha-1,4 linkage of starch molecule and digests the starch molecule into maltose, which is further degraded into glucose units. The autoprotection activity of pancreas is protected by the trypsin inhibitors, which are synthesized by acinar cells. Trypsin inhibitors are very active within pancreas and they become inactive only in the duodenal canal because of enterokinase activity. Likewise, pancreas has a neutralizing action of pancreatic juice, where acid chyme enters the intestine from the stomach and the pancreatic juice releases enormous amount of bicarbonate ions, which creates an alkaline environment and thereby neutralizes the acidic nature of chyme. Thus, this neutralizing effect protects the intestine from the destructive action of acid chyme. The phases of pancreatic secretions are regulated by nervous and hormonal factors. The secretion of pancreatic fluid occurs in three phases, namely the cephalic, gastric and intestinal phase, which corresponds with the stages of gastric secretions. Sensory inputs such as sight, smell, taste and mastication leads to the anticipation of food and initiate the secretion of pancreatic juice in the cephalic phase. When the impulse from the mouth or from the cerebral cortex reaches the vagal efferents, the vagal nerve endings release acetylcholine, which stimulates the pancreatic acinar cells to release the digestive enzymes. Thus, the cephalic phases of pancreatic secretion is under nervous control. In gastric phase, gastrin is secreted when food enters the stomach and is transported to pancreas through the blood and stimulates the secretion of pancreatic juice, which is rich in enzymes. During intestinal phase, chyme floods into the small intestine where eye cells in the duodenum and jejunum releases cholecystokinin into blood and binds to the receptors on pancreatic acinar cells and instructs them to secrete large quantities of digestive enzymes. Similarly, secretin is released by S cells of duodenum in response to entry of low pH stomach acid and primarily stimulates the production of aqueous sodium bicarbonate solution by pancreatic ductal cells. The generation of aqueous sodium bicarbonate solution washes out all the inactive pancreatic enzymes within the pancreatic duct into the duodenum where they are activated. The gastric and intestinal phases are controlled by the hormones. The endocrine functions of pancreas consist of islets cells that secrete hormones such as insulin and glucagon directly into the bloodstream, and these hormones in turn helps to control all blood sugar levels. Insulin released by the beta cells in the islets of Langerhans lowers the blood sugar level and moves glucose from the blood into muscles and other tissues so they can be used for energy. Pancreatic alpha cells secrete glucagon when there is a decrease in blood sugar, and its role is to break down glycogen into glucose in the liver. This glucose then enters the bloodstream in order to restore the level to normal. Thus, both the insulin and glucagon are implicated in modulating the carbohydrate metabolism. To conclude, in a differential organism containing various cell types, liver and pancreas has its own metabolic profile and specific biochemical functions to carry out most of its activities. Liver plays both anabolic and catabolic roles and furnishes all other organs with an appropriate mix of nutrients through the bloodstream. The liver, being the master gland, plays various roles in our digestive system, and proper channelization or care of liver does not lead to any ill effects. The role played by pancreas in digestive system is marvelous. Mainly, the exocrine functions and the endocrine functions were performed well by the pancreas. The digestive juice secreted by the exocrine glands plays an important role in the digestion of food materials, and the hormones produced by the endocrine system controls the carbohydrate metabolism and plays a vital role in the human body.